Good afternoon, everyone. I'd like to welcome you all. It's my pleasure to welcome you all to the annual meeting of the Cognitive Neuroscience Society. My name is Michael Anderson from the University of Cambridge, and I am the program chair for this year's meeting. And I'm really delighted to say that we have an incredible lineup for you guys this year. We have curated a collection of wonderful invited symposium sessions with topics ranging from consciousness to speech perception to task control, and also to memory, using a diversity of cutting-edge cognitive neuroscience methodologies from some of the leaders in the respective fields. I think you're going to find it to be an exciting couple of days and a very rewarding educational experience. We also have a plethora of member-sponsored or member-supplied symposia to keep you interested for the next several days. And of course, the all-important poster sessions, which is one of my favorite features of this conference, because that gives you the opportunity to get down and dirty and to talk to individual people about their science. And to meet some of the new up-and-coming scientists in our field. So please enjoy the poster sessions. We are privileged to also have an excellent slate of award addresses this year. We have Lynn Nadell giving us the George Miller Award, who's won the George Miller Prize. Professor Kia Nobra of Yale University, who's won the Distinguished Career Award, will be giving us a fantastic overview of the work that she's done. And we have, of course, a couple of young investigator awards to give. Peter Koch and Ella, I'm sorry, I'm not going to get this right. Ella Streim-Amit. Please correct me later if I've completely badgered that name. This conference is an incredibly complex thing to put together, and it would not be possible without the support of our sponsors. We had the V-Light Incorporated. They didn't ask for a shout out today, but thank you very much nonetheless. We also, I think we really have to acknowledge the extraordinary work of the different committees who've put together the scientific program, including my committee, the program committee, who did the invited sessions, the symposium committee, the poster committee, and the Young Investigator Awards Committee. Not to mention important people like Kate Trethewey, and Lisa Munoz, and Carrie Miller, who basically make everything work in this conference seamlessly, and so that hopefully you won't even notice the complexity that's involved in preparing this. Of course, I've left the best to last, which is that we have an absolutely wonderful keynote address queued up for you today, and which is the reason why we're all seated here. And it's my mission to get out of the speaker's way as quickly as possible. But but before I do, I have to tell you at least a bit about her. This is Professor Sheena Josselyn, Toronto's own Sheena Josselyn. She's a professor of psychology and physiology at the University of Toronto and a senior scientist at the Hospital for Sick Children here in Toronto, where she co-directs a laboratory, the Jocelyn Franklin Lab at Hospital for Sick Kids. She is, there's so many things that I could say about her. She sent me her CV, and I just like, oh my God, I just got this kind of sense of resignation that I am not going to be able to get through this. But she's, just to keep it brief, she's a fellow of the Royal Society of Canada, a senior fellow of the Canadian Institute of Advanced Research, and she's won quite a large number of awards, and I will leave it to you and the miracle that is Google to go look that up if you're interested in that. I'd rather focus a little bit on what she does, which is she focuses on how the brain encodes, stores, and uses information, and she does this primarily by studying mice, which is not the typical thing that we do in this society, but in my opinion, some of the most exciting science of memory that's being conducted today is being conducted at that level, and Sheena is at the vanguard of this kind of research. Using methods like engram cell labeling and optogenetics, is able to identify the neurons that are involved in the mouse encoding an experience into memory, either with respect to the amygdala or the hippocampus or pretty much any part of the brain involved in that experience can be labeled and identified. And with that in hand, you can study all of the important questions of memory. So the processes involved in forming a durable trace, the processes involved in causing traces to be forgotten. We have control over the memory trace. When I was an undergraduate, just to put things in perspective, this is a little bit of a depressing perspective, but when I was an undergraduate, it had only been something like 10 years since they developed the first successful animal model of hippocampus as a contributor to memory. And LTP had been discovered 10 years ago, and there was widespread resignation, like, yes, memory is in the brain. Yes, memory is stored by patterns of connections between neurons, but we're never going to know which ones. There's way too many out there for us to be able to identify that. It's too hard. And now we can, in part because of the kind of work that you're going to hear about today that Sheena is really involved in, is really applying in innovative ways. And so I think I'm going to make a little bit of a proclamation here. This is my opinion. The future of the science of memory lies in taking the innovations and discoveries of the work of people like Sheena and also Paul and Thomas Ryan and others doing this kind of work, taking that work and relating it to what we do here in this society at the level of systems neuroscience and at the level of human experience, being able to marry those two levels together and to fully vertically integrate how memory works. That's like the holy grail, and it just captures my imagination. And I really hope that it captures your imagination too. If even just a handful of people in this audience today are inspired by listening to the work that you're going to hear about and the comments that I've made here, to maybe graduate students starting their careers wondering what to work on, if a handful of you go on to try to draw that cross-level synthesis and do so successfully, I certainly will have earned my paycheck. So with having said that, I'd like to invite Sheena up on a stage and for me to get off.
Wow, well, thank you, Michael, for that lovely introduction. And it's amazing to be here today. To be in my hometown. My lab is just up the street, and it's, wow, a wonderful crowd out today. So I'm going to be talking about sort of my journey a little bit in science. And I know that some folks here are scientists, but some folks aren't scientists. So I'm trying to make this talk sort of digestible for everyone. So when I was getting ready for this talk, I was trying to come up with a theme for it. And this is a picture of, this is my daughter Charlotte, who is here today, and my husband Paul Franklin. And Charlotte is super excited because she is a huge Taylor Swift fan, and she has tickets for when Taylor Swift comes here in November. So this is a family outing to Skydome Rogers Center down the way, where Charlotte will be seeing Taylor in a few months, and these are actually the seats she's going to be sitting in, section 117. She got very good seats. So as a way of talking about the science I'm going to be presenting today, we're going to be talking about it in sort of Swifty terms so that everyone can understand and that everyone's engaged and excited by this. So I'm going to tell you a little bit about sort of the eras of the research that I'm going to be discussing today. And I'm very interested in, Michael said, in how the brain encodes, stores, uses information. We think that's the fundamental thing that a brain does. Our brain allows us to take things from the past and digest them and figure out what to do in the future. So this sort of how does the brain encode all this information is like a huge, big, fundamental question out there. And I'm going to start with a few lovely pictures. And I'm not sure if he is here today, but this is a picture of Dan Schachter. And he is a huge, as you probably know, memory researcher. He is the W.R. Keenan Jr. Professor of Psychology at Harvard University. He is amazingly smart. But here is a picture that Dan shared with me. So we're going to call this From the Vault, Taylor reference, of Dan way back in 1978 when he was a graduate student, again, just up the street in the Department of Psychology at the University of Toronto, and he was in Endel Tulving's lab. And Dan Schachter is a wonderful scientist, but even as a graduate student, he was pretty outstanding because here he is taking a sabbatical as a graduate student to go and research a very interesting figure in the history of memory. And this interesting figure in the history of memory is Richard Semon, and this is the book that Dan ended up making. He's here in Zurich reading letters from Richard Semon. And Richard Semon, he wrote this amazing book called Richard Semon, Forgotten Ideas, Neglected Pioneers, Richard Semon and the History of, and the Story of Memory. So Dan went back and traced the history of the term engram, which is the term I'm going to be talking about a lot today. And he went back in the history and talked about how science the sociology of science, in-groups and out-groups of science, who's remembered for what, who's forgotten. And he went back and really opened, at least my eyes, to this forgotten pioneer of memory. So this is a picture of Richard Semon, and what he did, mainly I'm going to be talking about today, are his contributions where he defined two terms. The first term is an engram, which is basically a memory trace. And he defined it in this wonderful language, the enduring, though primarily latent, in the irritable substance produced by a stimulus. Now, I'm assuming the irritable substance means the brain, but I'm not quite sure. So something happens to us, it leaves some impression on the brain that we can later recall, and that's how we can recall things. And then he also came up with this other definition called ekphory. He made the term up, and it's basically memory retrieval. And he defined it as awakening the engram out of its latent state into one of manifested activity. So these are really interesting and really, I think, still important terms to use. So I showed you a lovely picture of Dan Schachter that he was very, very nice to share with me. But here's another picture. So this is me. This is Paul Franklin and Stefan Kohler. And now we were also graduate students up the street at the University of Toronto in the psychology department. And we did not write a book as graduate students. I'm not sure if we read a book as graduate students, to be totally honest with you. But we did forge a really wonderful friendship, and we continue to be colleagues to this day, and we continue to collaborate and talk about memory. But we approach it from different perspectives. Michael talked about marrying different levels of thinking about memory. Well, I did that. I actually married Paul. So I am a molecular person. Paul is a bit more of a systems-y person. And Stefan, who I did not marry, but you never know, is more of a cognitive person. So Stefan has a position at the University of Western Ontario and is also appointed here at the Rotman, further up the street at Baycrest. So we get together and we like to discuss memory from different perspectives, and we like to write something about memory and see where we are in the field. And one of the things that I really liked that we did was we wrote this historical piece where we said, okay, these people really influenced our thinking, and why did they influence our thinking? And we went back in the history and started discussing who would make our list. So we called this piece the Heroes of the Engram. And so we went back in history and we defined who was really important to our thinking about memory and engrams. And we came up with a list. We started, of course, with Richard Semon, who defined the term engram, Carl Ashley, who searched for about 30 years of his career for an engram, Wilder Penfield, who maybe found an engram in a human brain, Donald Hebb, who talked about how cells fire together, wire together, and possibly become an engram. Brenda Milner, who of course is a memory hero to many of us and really defined the hippocampus as the place to look for memories. James McConnell, who studied this really interesting phenomena about memory in flatworms and how memory could possibly be transmitted from generation to generation in flatworms. And Richard Thompson, who looked for an engram in the cerebellum of rats. So this is sort of the eras of the engram that we are really interested in looking at. So when we were writing this piece, way back in 2017, we said, these people are so inspirational to us. They really are guiding our thinking. But we said, okay, none of them, it turns out, will ever win science's highest prize, the Nobel Prize. Now, why is that? Well, the majority of the people are dead. And one of the written rules of the Nobels is that they can't be awarded posthumously. Now, Brenda Milner, still very much alive, over 100 years old, but she violates the second unwritten rule of the Nobel Prize. Yes, she is a female. So let's break that down just a little bit more. So in our field, let's say physiology, medicine, chemistry, and physics, so the sort of sciencey bits of the Nobels, there have been 600 Nobels awarded in the sciences, and only 23 have gone to women. And the story is even bleaker for other historically excluded groups from science. Now, this slide is a little bit old. Let's add up our new Nobel winners. And again, we're sitting at around 3%. And I think this is, well, to quote Taylor Swift in her song, The Man, I feel as though I'm running as fast as I can. Would I get there quicker if I were a man? We'll let Taylor have the last word on that. So let's go back to Richard Semon. So throughout this, we have been sort of using this pretty archaic, old-fashioned definition of an engram, the irritable substance and these sort of enduring latent changes, What does that really mean? And people have been sort of saying, I don't like that definition. It doesn't say anything. And I love that definition because it doesn't say anything. It sort of gives you the general impression. But I think it's time that we updated the definition. So this is my version of the definition. So very much in the Taylor's version sense of things, I'm going to now be defining the engram as the ensemble of neurons active during an experience that are somehow important in the memory for that experience. So it's still vague, but it sort of gives you a sense that we're going to be looking at the level of the cell ensemble, a bunch of neurons that are come together. And then ekphory as the process by which external or internal retrieval cues reactivate the engram to produce memory retrieval. So this was our very first engram hero, and now I'm going to tell you a little bit about our second engram hero. So Carl Lashley. Now, he had a very interesting career, and when I first started as a scientist, I thought, he's sort of this sad cautionary tale. He worked for over 30 years of his career trying to find an engram in the brain of a rat. Can I find that bit of a rat brain that houses a specific memory? Of course, he made huge contributions in many other types of science and many other types of memory. He made a lot of contributions, but this was one question that he really wanted to answer. And how did he do it? Well, here's one of the experiments that he did. So he would train a rat to traverse this maze to reach this bit and get a bit of food. It's amazing. Rats can learn to do that. He would train them really, really well how to do that. And then he went in and lesioned out different bits of the rat's cortex, trying to find that one spot that held this memory for this maze, figuring that after he lesioned out this bit of the cortex, he would put the rat back on the maze. If the rat did really poorly, that means it was because Lashley had removed this part of the cortex. Well, it ended up, after about 30 years, he wrote this very influential paper called In Search of the Engram, where he basically concluded that the engram is everywhere in the cortex, but nowhere specifically. He could not find that one part of the brain, that one part of the cortex that held this maze memory. And at that point, this was published in 1950, studies examining the biological basis of an engram were largely abandoned. You could say that the engram had a bad reputation. So when I started thinking about engrams, I wanted to change things up a little bit. I thought that the concept was really important, that maybe we could make some headway if we changed up our task a little bit. So this is finding engrams, Sheena's version, and what I want to do here is change up the memory task. So here we're going to use a very simple Pavlovian type of training. So it's auditory threat conditioning, and it's super easy, and it's very easy to do in the lab. So we're going to take a tone that a mouse has never heard before and pair it with an aversive foot shock. So this is an aversive foot shock, but it's a little bit painful, it's a little bit scary, but it's like when you shuffle your feet on the carpet and then touch a doorknob. It's ay. It's nothing that's very long lasting. But mice, when you teach them this, they can remember it for a very long time. Now, how do we know they remember it? Well, we place them in a new place, and they're just roaming around, exploring. But then we play the tone, and if the mouse remembers, the last time I heard that tone, I got that really, quite nasty foot shock, the mouse will display this conditioned freezing response. So when a mouse is afraid, maybe you've seen one out in the wild, they adopt this crouch motionless posture referred to as freezing, and we can measure that very easily in the lab. So in this experiment, and many of the experiments I'm going to be talking about today, the way we measure memory is by saying how much time do mice spend freezing when they hear the tone. So we chose this task because it's super simple, super easy to do in the lab. It's one trial conditioning, and also because a lot of really pioneer work had shown us where to look in the brain for this type of memory. So we're going to be looking in the structure called the lateral nucleus of the amygdala. So this is a see-through brain of a mouse, and we're going to be looking at this part here, which is the lateral amygdala. Here's a coronal section, so an ear-to-ear section, and here's the lateral amygdala there. So we're going to try and look for an engram that holds this auditory threat memory in a mouse brain. Now, I mentioned that when I first started my career, I was like, Lashley is a very cautionary tale. But it turns out that I've not been at it quite 30 years, but I have been at it a good long time. So I started in this sort of looking at threat memories way back when I was a fresh-faced postdoc at Yale University in Mike Davis's lab, and in a collaboration with Eric Nestler. So what we did way back in the day was we were looking at how to enhance memory. So we used viral vectors, which was this really cool way of taking a bit of DNA that we wanted to put into cells in the brain. And we took a viral vector. Here we used a herpes simplex viral vector. So this is a herpes virus, and we take out everything that makes it herpes. And instead, we insert this gene of interest called CREB. Now, CREB had been, this gene, this protein, had been implicated in memory in a few other species. So what we can do here is take this viral vector expressing this protein called CREB, and we put a marker on it called GFP, green fluorescent protein, that we borrow from a jellyfish. It's what makes a jellyfish glow green. So we can see which cells we infect. So we're going to infuse this into the lateral amygdala of a rat here. And what we did is we found that it enhanced threat memory, auditory threat memory. And we were really excited about this because you can think of all these different manipulations that could potentially decrease a memory. But to enhance a memory, we thought was pretty special. And we were very excited about these findings until we looked at the brains of these rats. So here's an outline of our lateral amygdala, and it turns out that the infusions that I had done infected only about 10 to 15% of these principles, so these excitatory neurons in the lateral amygdala. So these neurons are the ones that are glowing green. And this really surprised us. So we had infected and we were increasing CREB levels in a really small population of cells in a really small area of the brain. And yet we see this enhanced memory, and we couldn't really figure it out. And we're sort of scratching our heads. Until we sort of started thinking a little bit, we sort of stood back and said, okay, what would we expect an engram to look like in the brain of a mouse or a rat? How would this be encoded? Would it be a very dense code? So every single neuron would be involved in this one memory. Or on the other end of the spectrum, would it be a very sparse code, like a grandmother cell? So one cell would hold all this information. And of course, it's probably likely to be a sparse code. So we wondered, well, maybe the reason why we're seeing this memory enhancement, even though we've infected this small population of neurons, is because these neurons are somehow preferentially recruited into this sparse engram supporting this memory. So that was what we tried to figure out from that point on. So here I'm going to just give you a schematic of how we're doing these studies. This is, again, our lateral amygdala. This blue is like all the different neurons. We're going to use our viral vector. We're going to overexpress CREB, and the neurons that we've overexpressed are going to be glowing green. So we wondered, are these neurons that overexpress CREB because they've been infected with our viral vectors, are they the ones that are part of the engram encoding this memory? And so we wondered how we could figure this out. And we thought that one way we could look at this would be to test the mice and figure that those neurons that are active right immediately following the test are the neurons that are part of the engram. So we identify these neurons based on this activity-dependent marker. And what we found is that neurons that we overexpressed CREB in were really, really likely to be part of these engram neurons, so these neurons that were active following our memory test, way higher than would be predicted by chance. So we are very excited by that, but CREB is a transcription factor, and it does many, many different things. So we said, well, what is it about CREB that makes neurons with high levels of CREB more likely to be recruited into the memory trace? So we did a bunch of experiments where we either overexpressed CREB, or we overexpressed potassium channels, or we used these optogenetic or chemogenetic tricks. And the one common denominator of all these manipulations is that they increase the excitability of these neurons right before the training. And we found that indeed, all these ways of increasing excitability, these neurons seem to be preferentially allocated into the engram supporting this memory. So then there we go. So we thought that, okay, neurons that are allocated to a sparsely encoded engram are allocated based on relative excitability around the time of training. So we came up with this model that these three neurons, they start out as equal. Any neuron can grow up to become an engram neuron, but this neuron becomes an engram neuron because it's more excitable than its neighbors. So we think that neurons with relatively increased excitability around the time of training win this competition. But how do we show that? This is all very correlational. This neuron tends to be like this, and this neuron tends to be like that. So what we really want to do is the updated Lashley experiment. So if we think this neuron is really important in the engram supporting this memory, the thing to do would be to somehow target just this neuron after we've trained an animal on something. And if we think that this neuron isn't really important, it's not important in this memory, we could target this neuron in a different group of mice and see what happens to memory. And this is exactly what we did in the next experiment. So we're going to have two groups of mice. In one group of mice, we're going to overexpress CREB like we always do in a small, sparse population of cells. In the other group, we're only going to express GFP. We're only going to mark the cell. We're not going to do anything to its activity or excitability. We're going to train a mouse. We're going to test the mouse as normal. And when we test the mouse as normal, we see normal levels of memory and similar levels of memory. Here's how we test memory, the percentage of time that mice spend freezing to the tone, equal in both groups, which is very cool. But then we're going to use this really neat genetic trick, which allows us to kill just these neurons that we've infected. So in our CREB group, we're going to kill those neurons that were overexpressing CREB, so neurons we think are really important in the engram. And in the other group, we're going to kill just cells that are GFP expressing. So they just glow green. We don't think they're really important in the engram. And we're going to test the memory again. And when we do that, we see a memory deficit when we kill the neurons that were overexpressing CREB, suggesting indeed they are very important in the engram supporting this memory. In our control group, if we kill just a sparse population of neurons, we don't see a memory deficit. So you could say that we were able to make these mice fearless, not quite fearless, but at least we could decrease their memory. So this was one of the first big papers that came out of my lab, and I remember the day it came out all too well. Yes. It was March 9th, 2009, and I was very pregnant up at Mount Sinai Hospital. And that's a hospital right across the street from my hospital. And we were so excited about this finding coming out, and the person who had actually done the majority of this work didn't feel comfortable talking about it. But I was so excited about all the hard work he'd done on this that I wanted to tell everybody about it. So here I am actually doing press while in labor. And this is the cover of the Toronto Star the next day. They say, "Science, a step closer to the spotless mind." And in it, they say, Sheena Josselyn, a sick kid scientist, politely, I am Canadian, declined an interview request from the Star because she had just gone into labor. And here we go. There is Charlotte at her very first conference, and now she towers over me. So this is Asim Rashid, and he is, as you can see, a very hardworking research associate in the lab. Here he is channeling his inner Taylor Swift, but without the red bottom on the boots because he's a research associate. And he said, these experiments are very, very cool, but killing cells, that's kind of an old-fashioned technique. Can't we be a bit more bejeweled in all of this? So what he's going to do is use one of these modern neuroscience techniques that Michael was talking about before, and it's very cool. We now have the ability to control the excitability of cells by using light. So what we do is we borrow genes from algae, and we shine different wavelengths of light into the brains of mice, and we can really control their activity. And this has given us unprecedented ways to explore memory, as well as other things in the brains of mice. So what Asim is going to do is use this very cool viral vector that we made that expresses two different flavors of these optogenetic constructs. So each flavor is excited by different colors of light. So if we shine a blue light, we hit channel rhodopsin, which is an excitatory opsin. So here's a neuron, and it's firing, 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 firing. So we can make a neuron very excitable when we shine a blue light on it. But the same bunch of neurons, if we shine a red light, we hit this other flavor of opsin, halorhodopsin, and it silences these neurons. So these neurons no longer fire. So he's going to use this really cool turn on, turn off activity of these cells to do a similar sort of experiment. So what he's going to do is take this viral vector that expresses, again, both channel rhodopsin and halorhodopsin. He's going to sparsely infect a small population of neurons in the lateral amygdala. And in one group, he's going to shine the blue light to try and excite them right before the time of training, trying to allocate them into the engram supporting the memory. He's going to train mice as normal and test them as normal, too. But he's going to test them actually twice. First time, he's going to test them normally, and second time, he's going to test them while he plays the red light. So he's just going to silence those cells that he's tried to allocate into the engram. So he's going to silence these exact same cells, and everything is counterbalanced. And when he does that, he sees first, this is what happens with the open bar is what happens with the red light off. So just testing normally, the mice are showing high levels of memory. Then he waits a couple minutes, he plays the tone again. This time he's shining the red light, he's silencing those cells he's tried to allocate, and we see a memory deficit. And importantly, when he does this in control mice, so exactly the same thing, only he's not going to excite these cells right before the time of training. They're just going to be a random small population of cells we don't think are very important in the engram supporting this memory, because when he turns them off when he turns them off during the test, he sees no effect whatsoever. So only when we turn off or kill these neurons that he's tried to allocate into the engram do we see a memory deficit. So this we thought was pretty cool experimental evidence that these neurons are somehow necessary for memory. And this is Sungmo Park, Emily Kramer, and Annalise Horn. These are two graduate students and a postdoc in the lab, and they said this is very cool, suggesting that these neurons are necessary for the memory. And they said, but are they also sufficient? So they're going to try and do this very cool experiment. So they are going to have channel rhodopsin, so this optogenetic construct, when you hit it with blue light, it increases the excitability of these neurons, and they're going to try and allocate these neurons into the engram supporting this memory. Now, during our memory test, what we usually do is give the mouse a really good memory cue, so we play the tone. And we think that the tone creates memory retrieval by reactivating the engram. So they want to do a really cool experiment where they just say, okay, what happens if we take out the middle person here? What if instead of having the tone reactivate the engram, what if we do it optogenetically? So what they're going to have is they're going to place mice in a novel context. They're going to see what freezing looks like. And then they're going to try and ask, what happens if we optogenetically reactivate the engram? Can we get the mouse to behave as if this mouse is recalling this threatening, fearful memory? So this sort of sounds like what Simon described as ekphory, awakening the engram out of its latent state into one of manifested activity. Now, it could be that, if we want to get all Nostradamus-y, we could say, Simon foresaw optogenetics. Maybe he's in line for a prize, but probably not. So this is the data. This is what happened. So when they place the mouse, just in a novel context, the mouse shows very little freezing. Then they turn on the blue light, try and reactivate this engram supporting this threat memory, and the mouse freezes. They turn the light off, the mouse freezes a little less, turn it back on, it starts freezing again. Now, this only happens if we train the mouse. If we don't train the mouse and we just shine a light to activate a small population of cells in the amygdala, the mouse doesn't freeze. The mouse has to have this training. We have to allocate these neurons. Now, is there a difference between a naturally recalled memory and this artificially recalled memory? Probably, and we're explaining that right now. But the fact that we see anything that remotely appears to be memory retrieval is very cool. So, this is Taylor. And here she is in her mugshot for No Body, No Crime. This is Liz Huang and John Howland, and these are their mugshots. And they have done a very, very serious crime for which they were sentenced to spend some time in my lab. So what they asked was, okay, this is really cool. You've looked at all these sort of threatening type memories. What about an appetitive memory? Do we see an engram, and does it seem to form in the same way? So what their task was to find a procedure where we could induce one trial appetitive learning. That's what we do in our auditory threat experiments. It's one trial. We pair tone with a foot shock. Can we find something as rewarding to a mouse as a foot shock is aversive? So here's what they did. So they put the mouse, they asked the mouse to skip lunch, and they gave it some very yummy lab chow. But that wasn't enough to give it a very strong one-trial memory. So then they said, everyone likes chocolate. I'm sure mice like chocolate. Maybe we can get the mouse to remember after this yummy chocolate treat. That didn't work. So then they did chocolate milkshake, and we used chocolate milkshake as a reward in some of our other tasks, but that wasn't enough to form a very strong one-trial memory. So they were sort of looking around trying to find, well, what is really rewarding to a mouse? And, no judgment here, judgment-free zone. But it turns out that mice really, really like is cocaine. Yes, so we're going to train this mouse with one dose of cocaine, and we're going to do it in a slightly different way. So we have this very simple apparatus with this guillotine door, and there's two rooms in this box, and they have different cues in the rooms. And what we do on the very first day, when a mouse is drug-free, nothing's going on, we take out the guillotine door and we say to the mouse, where do you like to spend your time? And the two rooms are pretty equal, and mice spend about equal amounts of time in each side. Then on the training day, we put in the guillotine door. In the morning, we give the animal a simple IP dose of saline. It doesn't do anything. We confine them to one side. In the afternoon, we give them a single dose of cocaine and confine them to the other side. Then the next day, the mice are drug-free. We get rid of the guillotine door, and we say, where do you like to spend your time? And mice who really, really like something will spend a lot of time in that area of the apparatus where they previously had that amazing experience. And it turns out that mice really like cocaine. So this is their preference before the training, and this is their preference after their training. So now we have a very different type of appetitive memory, very different from what we did with our auditory threat conditioning, but still one trial appetitive memory. So we're going to see what happens if we can allocate neurons just like we did with our aversive memory. So we're going to, again, infect a sparse population of cells. They're either going to overexpress CREB or use optogenetic actuators. So we're going to increase the excitability of these cells. And it's just like what we saw in our aversive memory, we are able to bias the inclusion of these cells into an engram supporting this cocaine cue memory. And what's really cool is that these allocated neurons are necessary for subsequent expression of this memory. So if we kill these neurons, if we turn them off, mice no longer show us this very strong place preference. And once the mice have learned this, we can then go on and these cells become very rewarding in and of themselves. So we see that increasing the excitability of a sparse random population of individual lateral amygdala principal neurons can bias their allocation into either a threat memory or a rewarding memory. And this suggests to us that at least these, in the LA, these principal neurons are not irreversibly tuned or in other ways hardwired to become part of an aversive or an appetitive stimulus. They can grow up to be whatever kind of neuron they want to be. So this is where I started sort of my career in this field with Stefan and Paul. And, you know, we never go out of style because we still get together. And this is us together at some point in 2019. And in 2019, we decided that we would write a review talking about the neurobiological foundation of memory retrieval. So memory retrieval is very much studied in cognitive neuroscience, but a little bit less so in the sort of biological neuroscience, where people tend to really focus on memory encoding. And Stefan said, well, I think there's a lot of room to look at memory retrieval in the basic neurobiology. So again, we're going to really focus now on ecphory, this process of memory retrieval. And we're going to turn back to this really important finding from Endel Tulving. And Endel, as you know, was a part of our department at the University of Toronto until he passed away very recently. And he produced a number of really important findings. And the finding that we're going to follow up here is talking about the availability versus the accessibility of memory. So he came up with this hypothesis called the encoding specificity and retrieval process of episodic memory, which basically means that you remember something better when the conditions between the encoding and retrieval are really, really similar. And this is called the encoding specificity hypothesis. And this is best illustrated by a really fun study from Godin and Badley in 1975. And instead of giving the Taylor twist here, because we're in Canada, I'm going to give it the Canadian twist. So this, as you all know, this is Justin Bieber. And he is wearing traditional Canadian garb, although I don't think that's a traditional Canadian greeting. So in this sort of study, what they're going to do is they're going to train Justin on a list of words. But you might not know this, but Justin is actually an accomplished scuba diver. Here he is underwater. And they can train him on a list of words while he's underwater as well. So these are the training conditions. He's either going to be on dry land or underwater, and then we're going to test him either on dry land or underwater. So if we do dry land, and underwater, we call that a match. That's high congruence between the training and the retrieval conditions. The encoding specificity hypothesis says that's a really good way of recalling a memory. But if we do the mismatch conditions, if we train Justin underwater and get him to recall the list on land, there's not a high congruence, and this leads to memory retrieval failures because it's a mismatch. So this is the sort of the nuts and bolts of the encoding specificity hypothesis. The information is still there in the brain, but sometimes we have difficulties accessing the information. So we thought it would be really fun to, if we think memories are encoded in engrams, to update this hypothesis a little bit. So we came up with the engram encoding specificity hypothesis, which we thought said that high congruence between encoding and retrieval conditions contributes to successful memory recall because it efficiently activates the supporting engram. So the engram is still there at times. It's just sometimes we have difficulty accessing it by the retrieval cues. So here's how we can do these studies. So we encode, the mouse encodes an animal in one condition. If we ask it to retrieve it in a certainly, a very similar condition, that's a match. We get good activation of the engram, we get good memory recall. If if we give it a very different type of recall experience, then we wouldn't get a very good, we wouldn't get a very efficient activation of the engram, and we wouldn't get very good memory recall. Even though the engram is still there and available, it's just not being accessed by this retrieval cue. So we wanted to explore this a little bit more. This is Jung Hoon, Ying Wang, and Andrew Mokley. They are 3 students in the lab, and they did a bunch of experiments where they changed the encoding versus retrieval conditions in mice using the same auditory threat conditioning paradigm, and they looked for engram reactivation and memory retrieval. So we tried several different ways of changing up encoding versus retrieval. And one of my favorite ways is exemplified here. This is, because we're in Toronto, this is Drake. And Drake has his own line of whiskey. I think it was a professor, or maybe it was my mom, it probably was my mom, told me when I was younger that if you study for a test drunk, you should take the test drunk, of course. So we are going to test this in mice looking at engram reactivation. So we are not going to use single malt. We're just going to use good old lab ethanol. So we're going to do just that experiment. We're going to have 4 groups of mice. Right before the time of training, one group is going to get an IP injection of ethanol, another one's going to get saline. We're going to train them like we typically do, only this time we're going to do 2 tone shock pairings. And then we're going to test them either while they, either after they've been given ethanol or saline, and we're going to test their memory. But we're going to do a trick where we can sort of tag which neurons are active at the time of training. We call this engram tagging, and we're going to use a way of assessing how well these engram cells are reactivated at test. So the cells that are engram cells are going to be labeled in green, and then we're going to look for the cells that are active, the engram reactivation at the time of test when they are red. And first, let's look at our behavioral findings. So the stippled bars are the matched conditions. So this group of mice had saline before training and saline before testing, and no big surprise, they have very good memory. But what's really cool is that the group of mice that had ethanol before training and ethanol before testing also had really good memory. The 2 groups that had the mismatch conditions, saline ethanol and ethanol saline, did not have very good memory recall. Very consistent with the encoding specificity hypothesis, we changed the retrieval environment. The cue could no longer access the engram. Is this true? Yes, when we look at the engram reactivation, we see higher engram reactivation in our matching conditions and lower engram reactivation in our mismatch conditions. And when we look at individual mice, we see a correlation between the degree of engram activation and the degree of memory recall. So this is one experiment where we tried to change up the encoding versus retrieval conditions using very, very different types of cues. We've done this using different strategies to tag an engram. We've looked at different ways of changing up match or mismatch the encoding and versus retrieval conditions. And over all these experiments, these individual data points are all individual mice. We find that the engram size in the lateral amygdala is pretty constant. The number of neurons active during a test are fairly constant. But what's really cool is over all these different ways of changing up the encoding versus retrieval conditions, we see this really nice correlation between the overall engram reactivation in the lateral amygdala and the amount of freezing, so our memory retrieval in these mice. What we've done in most of our experiments is to use all these kind of wild and crazy techniques to change the activity of a population of neurons. So we infect these neurons with viral vectors, we put in optogenetics, we put in little light pulses into the brains of these mice, so all these different things. But what we really want to know is, does this process occur at all endogenously? So this is Chen Yan, and he was a graduate student in my lab. And what he wanted to do was to actually watch cells as mice learned and recalled a memory, to try and look to see if these neurons that are more excitable right before a mouse is trained on something become engram neurons. So there's different really cool ways of doing that, and there's a lot of wonderful endoscopes that have been made that allow you to look at the activity of individual neurons. So you put a little lens into the brains of a mouse, you put in an activity indicator into the brains of these mice, and you can make an endoscope. And Denise Kai is right here in the front row, and she and her colleagues have made this amazing open source one. In my lab, this was Chen, and he made his own endoscope, and we call it a Chendoscope. So here it is. Here's a Chendoscope. It's really easy 3D printed, and it costs about $2 Canadian. So this is Chen Yan. He has now left my lab. He graduated, and he now works at Google DeepMind, and that's in London. And if you know anything about the geography of London, you might know why the entire family is really excited to visit him. So he works here at DeepMind. Right across the street is King's Cross Station, and that's where platform 9 3/4, and we catch the train to go to Hogwarts. So this is an experiment done by Andrew Mokley, Adam Ramsaran, Lena Tran, and Alex Jacob, and it's a collaboration between my lab, Paul Franklin's lab, and Blake Richards' lab at McGill. And what we do is we have these mice that are only expressing a calcium indicator, so that lets us read out the activity of individual neurons. And again, they're wearing a Chendoscope. And what we are going to do is train these, now we're going to look at dorsal CA1 hippocampal neurons. We're going to change up our task just a little bit. We're going to have context conditioning. So we're going to place the mouse in a context, and then we give it 3 foot shocks, and then we test it 24 hours later. And again, what we really want to know are these neurons that are endogenously more active, are they more likely to be allocated into the engrams supporting this context threat memory? So what we're going to do is record the activity of these neurons immediately beforehand while the mouse is in the home cage, 3 hours beforehand, and 24 hours beforehand. And importantly, we don't tell the mouse what's coming up, so they have no way of knowing. We're just going to record these neurons. We're also going to record what happens to these neurons 24 hours later in a different context. So first, we're going to identify engram neurons as those neurons that are more active during the test. And when we do this, we find about 7% of the neurons that we image of these 1,500 neurons that we image are classified as engram neurons. The rest of the neurons are going to be non-engram neurons. So what are we going to do? We're going to have our 2 groups of neurons, engram neurons and non-engram neurons, based on their activity during the test. We're going to be able to trace back and see which of these neurons were more active during which session. So if we look 24 hours in the home cage, we see absolutely no difference in the activity between these engram neurons and their non-engram neighbors. But during the test session, during the training session, as would be predicted, we see higher levels of activity of these engram neurons. But what's really cool is even immediately beforehand or 3 hours beforehand, the mouse does not know what's coming. We see these neurons that go on to become engram neurons are more excited than their neighbors, and they're not if we test them in a different context 24 hours later. And we can do this using multiple ways of labeling engram neurons. So these are just a few of our efforts sort of looking at engrams and memories, and it's been a really fun time in the lab. And I just want to highlight that certainly in my little area of neuroscience, with the aid of new tools and really wonderful old theory, science fiction is becoming science fact. And these are 3 movies about memory and different aspects of memory, and we're sort of getting a little bit of data that suggests, maybe at some point. But I would just want to point out, because, fire. Oh, I shouldn't say that, right? Free speech, but you're not allowed to say that. What, do we, Oh, don't, everyone stay seated, I'm taking names. So, this is a uniquely, these movies are uniquely Toronto. So Eternal Sunshine of the Spotless Mind, Jim Carrey was born in Scarborough just down the hall. Total Recall, where they talk about memory retrieval, the original with Arnold, was filmed here in Toronto. And Inception, apparently where they implant memories, not a huge Toronto connection, but Elliot Page, who acted in it, was born in the east coast of Canada and lived in Toronto for a few months. So with that, I just want to thank my funders and point out this amazing symposium that's going to be happening tomorrow. So it's going to be memory engrams and their implications for human memory, and it's going to have Denise Kai, Paul Franklin, Inbal Goshen, and Thomas Ryan. So don't stay out too late tonight, but do stay out really late the next night, because Pavlov's dogs are going to be playing at Adelaide Hall. And of course, later on Friday, next Friday, Taylor Swift has a new album dropping, and I know my house is hosting a listening party and sleepover, and you are all invited. Again, thank my husband and my wonderful daughter. And this is the eras of the Josselyn Franklin Lab. We take a selfie every year, every few years, and this is all of them. So thank you very much for your time and attention. And I think we have a reception outside. So everyone, let's go outside and see what this is all about.